My name is Josh Leeman. I work at uniQure in business development and lead search and evaluation. Thank you ahead of time for bearing with me. I'm pinch hitting for a colleague who couldn't make it, so we'll see how this goes. First off, thanks to the organizers of the meeting on the Mesa for giving uniQure an opportunity to speak today and share with you a little bit about where we are in our strategy moving forward. I'll be making some forward-looking statements today, all of which are outlined in our SEC filings and company disclosures on our website, uniQure.com. So to start, I figured it'd be the best place would be to go back 23 years to the founding of the company and share with you what our mission was. And that was to deliver curative, one-time administered genomic therapies and medicines that transform the lives of patients. We're not looking to modify symptoms or gain marginal improvements. Our aim is to significantly modify the course of serious disease by harnessing the power of genetics. And so over the past 23 years, this really hasn't changed. And some of the things we've done recently, we feel have enabled us more than ever to deliver on this mission. Our strategy moving forward has been further enabled by the deal we announced in 2020 with CSL, and we've also reimagined the focus on where we can best direct our platform. That is the indications we can address, the cargo we can deliver, and how we can most efficiently manufacture our products in a cost-effective, reliable manner. So highly valuable to uniQure is our platform. We believe it serves as the engine for developing transformative medicines, rather. Certainly, AAV gene therapy can be used broadly, but we've chosen to focus in three main areas: CNS disorders, liver-directed disorders, and cardiac and muscle diseases. Over the last 20 years, we've invested heavily on understanding the unmet medical needs encountered by these patients and how we can best develop transformative medicines to address these issues. Core to our strategy is to continue to leverage this AAV gene therapy engine and the technological platforms we've established to innovate in these key areas. So in contrast to the competitive landscape we encountered in 1998, there are now hundreds of gene therapy companies out there, with more and more being announced every day. However, along this path, we believe that uniQure remains differentiated as both an innovator and leader in the space. We were the first to gain regulatory approval for a gene therapy. We were the first to have a commercially licensed gene therapy manufacturing facility. And with our Ph.D. program, we were the first to deliver gene silencing therapy to the clinic. Our AAV5-based platform in hemophilia B was the first to demonstrate positive clinical data in patients with pre-existing neutralizing antibodies. uniQure's leadership in the field has also been demonstrated through deep research and manufacturing experience. Over the past 15 years, we've continued to invest in our commercial cGMP manufacturing. We've used those products to dose 8 different clinical trials, over 100 different patients, and we've seen up to 10 years of follow-up. This can only really have been done with world-class researchers from the United States and Europe, experts in CMC and manufacturing, and all of which have extreme technical expertise. It's this deep experience that's helped us establish a leading portfolio of IP and know-how covering manufacturing processes and methods, as well as novel tools and enabling technologies. One of the best well-known successes at uniQure has been our hemophilia B story. It started in 2008 when we in-licensed the Factor IX gene therapy from St. Jude's, and by 2015 we had initiated a clinical stage phase 1/2 program, AMT-060. But by 2017, most in the field thought the program was dead. But given the confidence we had in the platform and AAV5, we slightly modified our approach, incorporated the Padua variant, and were able to convince regulatory agents that this was highly comparable. So by 2018, we initiated a phase 2B comparability study, followed soon thereafter by our phase 3 HOPE-B study. Last year, we were able to fully enroll and had completed dosing of all 54 patients in the trial. And earlier this year, we announced 1-year follow-up of each patient. And then last year, we announced a $2 billion out-licensing and collaboration agreement with CSL. This was critical for us because CSL is a leader in the global leader in hematology with a commercial footprint in 80 countries. This deal represents a huge opportunity for uniQure to grow our pipeline and platform even further. Our Huntington's disease program is also emblematic of our innovative nature. We spent 6 years developing a technology called uniQure, which is our proprietary gene knockdown technology. And with this platform, we were the first to enter a gene therapy trial in Huntington's disease, which we know is a disease of extremely high unmet medical need, with 100,000 patients being affected in the United States and EU alone. Earlier this year, we initiated our phase 1/2 clinical trial in HD. It's double-blinded, sham-controlled, and was strongly endorsed by the FDA. We believe this would expedite our path to registration. And then later this year, we hope to have some very, very early biomarker and safety data in the first 4 patients dosed. And then in combination with the EU trial that we will start soon, we hope to have additional data readouts over the next couple of years. So following the CSL deal for AMT-061, we are really highly focused on leveraging these financial resources and expanding the pipeline, particularly those of clinical stage or near clinical stage. Currently, we have 2 clinical stage programs, Hem B as I noted, and HD as I noted, both of which we believe are transformative to patients, or will be transformative to patients. But looking forward, by 2026, we hope to have 8 to 12 commercial and clinical stage programs with a full roster of next-gen preclinical stage programs that we think could also be transformative. Now, this is an ambitious goal, but attainable given the confidence we have in our platform. We realize that to maintain a healthy pipeline, that a level of attrition is required. And to ensure the highest level of R&D efficiency and effectiveness, we are working to reduce our timelines by leveraging a more modular product development platform and selecting new programs that have more balanced risk-reward profiles. So in terms of how we balance this risk-reward, we've really sat down and refocused how we evaluate and select new programs and targets, and now view this as a key part of our DNA. Our aim is to always develop gene therapies that can be best in class or first in class, and if we can't envision a program being that, we won't waste the time or money. Additionally, if there's emerging data or the competitive landscape changes, we are secure that we are able to move on. And I think our past actions in hemophilia A and B support that. Shown below here are a couple other critical areas that we use to evaluate new targets and diseases. We focus on indications where there's a large unmet medical need, work on targets that are validated in humans either pharmacologically or through human genetics. We look for safe targets to complement our very safe delivery platform. And we try to ensure that we can build programs where we can evaluate it preclinically and that transfers over into what we can expect in the clinical setting. And of course, we want to ensure that we can actually run clinical trials. Lastly, where at all possible, we like to leverage technologies that we are confident in. This is why you've probably seen that we have used AAV as a workhorse in many of the programs that have come through uniQure. And as you'll see in a few slides, we believe that these characteristics have carried through to all of the programs in our pipeline. So enabling technologies and platforms are essential to our strategy. We view these in 2 broad buckets. The first bucket has to deal with where and how we deliver the DNA material. Much of the AAV gene therapy programs that we've seen over the last 2 decades have dealt with first-generation capsids and standard delivery technologies. So today at uniQure, we've established and continued to develop novel capsids, next-generation promoters, improved formulations, and optimized administration technologies, all of which are aimed to answer critical questions around potent expression and improved efficacy, how we can better target delivery to specific cells and enhance biodistribution and safety, how do we achieve broader transduction of an entire organ, And lastly, and very importantly, how do we achieve redosing to enable dose titration and durable efficacy in pediatric populations? The second category has to deal with what we deliver, the DNA constructs themselves. So most gene therapies we can think of deal with replacing missing proteins and enzymes. At uniQure, we're constantly thinking of new ways to expand the pipeline and broaden the applicability to more patients. So to that end, we've used technologies to reduce gene expression. I mentioned that earlier, called uniQure with Ph.D. And we've taken these learnings and we've applied it to a new technology we call QURE, which reduces and reestablishes expression of perhaps a different gene. And then most recently, we have a vectorized antibody platform called AbQURE. Each of these programs significantly broaden the applicability of AAV gene therapy. So one of the things we're now looking at is we're taking what we've learned from the new target and disease area evaluation process and the various enabling technologies I've just shared to look at much larger indications where there's massive unmet need, and we think we can maximize our efforts to really address the most patients possible. So in June, during our R&D day, we announced 3 such programs, one in Parkinson's disease, one in Alzheimer's disease, and one in ALS, all of which affect tens of thousands of patients. And we believe that moving from ultra-orphan or orphan diseases to the more common ones is really the next chapter in AAV gene therapy. So this is now built up to our pipeline. This is how it looks today. At the top is our most advanced program, phase 3 program in hemophilia B. That's now been outlicensed to CSL. At the bottom, you can see we have a partnership with BMS on 4 undisclosed targets. And then internally, our internal pipeline consists of programs that are liver-directed, as well as those that are CNS, including HD, Parkinson's, ALS, Alzheimer's. And we recently announced the acquisition of QURE, a French company, and their program in temporal lobe epilepsy. So a final critical aspect of uniQure's leadership in AAV gene therapy concerns how we manufacture these therapies. We've always viewed manufacturing as existential. And first, the manufacturing process and methods are inextricably linked to the product itself. Second, it's currently quite expensive to manufacture them at commercial doses. And it's probably currently very difficult to address these large programs I just mentioned in a cost-effective manner. Lastly, it's quite difficult to manufacture these AAV gene therapies as biologic materials are notoriously inherently variable, difficult to scale, and challenging to characterize. So over the past 15 years, we continue to invest and innovate towards a robust, highly optimized manufacturing platform that's modular across programs and can be scaled efficiently. We use the same platform from the earliest stage research programs all the way through to commercialization of a product. This maximizes the consistency we see and the comparability through development. So in order to address these larger indications, we're now working on plans to scale the platform even further, using bioreactors that can generate thousands of liters of drug substance in an even more cost-effective manner. In order to maintain our manufacturing leadership, to support CSL's commercialization in hemophilia B, and to expand our pipeline, we will continue to invest in our capability infrastructure. We recently announced initiation of a second manufacturing site in Amsterdam. This will be capable of making 500-liter cGMP material by 2022. And then at our home base in Lexington, Massachusetts, we are building out a pilot process to complement our in-place cGMP process, as well as to help with the new research programs we'll be announcing next year. This will make tech transfers even more easy and efficient. So going forward, we're really focused on several different things. One is hemophilia B, of course. We did close the commercialization licensing agreement with CSL. We will support them through manufacturing. We presented the 52-week HOPE-B data from the pivotal trial. We're on track to announce the 78-week top-line data and prepare for regulatory filings and marketing approval. In Huntington's disease, we have now enrolled the first patient in our second dose cohort for the phase 1/2 trial in the United States. We're on track to begin our enrollment in Europe in the phase 1b/2 trial. And as stated earlier, we hope to have some very, very early preliminary biomarker and safety data. And we continue to evaluate new disease areas targets and develop enabling tools and platforms as we expand our pipeline and capabilities. Thank you for bearing with me, and I'm happy to talk afterwards if you're interested.